Hello, in today's class, we'll talk about gene expression regulation in prokaryotes and also in eukaryotes. So, first part of the lecture would be gene expression regulation in prokaryotes, and in that regard, we would talk about Lac Operon and Trip Operon, and the later part of this lecture would deal with eukaryotic gene expression and how they are complexly regulated, how are the histone modifications, how are the chromatin is remodeled, how the chromatin landscape is changed, and all those things. So let's just begin with our lecture with a module of prokaryotic gene regulation. We talk about prokaryotic gene regulation, and under this module we'll talk about two things. One is the Lac Operon, and second is the Trip Operon. So let's just begin one by one. So first we talk about Lac Operon. So here we have to ask ourselves what is Lac and what is an operon. So simply Lac means lactose and operon means multiple genes are controlled under one promoter. So definitely the Lac means lactose and operon simply means a multi-gene system regulated by one promoter. So this whole thing has to somehow deal with regulating the gene expression, which might be useful for lactose metabolism and all. The question is, why does bacteria need lactose? Definitely for nutrition. And what is the preferred nutrition for a bacteria? So bacteria, especially E. coli, prefers glucose. So a bacteria and E. coli would prefer glucose over lactose. So they prefer glucose more because for them glucose is easy to digest versus the lactose, because lactose is a little bit complicated than the glucose. So if the bacteria, the E. coli, is given a choice between glucose and lactose, they would always choose glucose as their nutrient source. But think of a situation when only lactose is present in the environment but no glucose. Then what? Then it need to survive, and in order to survive it need to metabolize the lactose and in order to produce energy and other necessary metabolic processes. So they need a lactose digestion system or lactose metabolism system, and that is regulated by Lac Operon. So Lac Operon simply give rise to expression of the genes which are important for lactose metabolism. Now definitely we have to understand whether in a normal situation when glucose is high, what is the status of the Lac Operon. So when glucose is high, it has been seen that lactose operon is off. In other sense, when glucose is low but lactose is high, then Lac Operon gene expression is on. So we draw a bacteria, a big bacteria here, to look at what is happening inside. So inside the bacteria, we definitely have our bacterial chromosome, and a part of that bacterial chromosome has this Lac Operon genes. So here is our Lac Operon genes. It has Lac C, it has Lac Y, it has Lac A genes. All of these three structural genes are under the Lac promoter, or otherwise known as Lac P. In this whole situation, there is another player. Another player is in this whole situation is Lac repressor, which is a product of the Lac I gene. Now this Lac I gene is under the control of a separate promoter. Okay, now we look at what all of these genes would give rise to if it is expressed. If these genes are expressed, then Lac C would give rise to an enzyme known as beta galactosidase. Now, beta galactosidase is an important component which converts lactose to its simpler form, glucose and galactose. In some rare cases, beta galactosidase can also convert lactose into allolactose. Now, allolactose is one of the isomers of lactose, and in a moment we would look at what is the function of allolactose in Lac Operon. However, the Lac Y gene gives rise to a permease protein known as lactose permease. And Lac A gene gives rise to galactoside transacetylase. So these are the three important products that are given by these lactose operon genes. Now, lactose permease is important because lactose permease is the thing which allow lactose entry into the cell. So for lactose entry, lactose permease is very important. And Lac Z is important because Lac Z converts the lactose which is entered by lactose permease and make glucose and galactose, which are the relatively easily digestible things for the bacteria. Now, what happens, we would look in a bigger picture that how these things happen. I mean, how this gene expression is regulated under different availability of different nutrients. So we take an example of a simple example where glucose is present. So glucose is present in very high levels in the environment. Now in this situation, imagine that glucose is the preferred food for the E. coli, and E. coli won't like to waste its energy to metabolize lactose and then break it down into glucose and then utilize that. Instead, glucose is readily available in the environment. Take the glucose, use it for its metabolic purposes. So that's why lactose operon gene should be turned off, or the lactose metabolism should be turned off at that point of time when glucose is high in the environment. In this context, let us look at what is happening at the gene expression from the Lac Operon. So we draw the Lac Operon. So we definitely draw the Lac Operon. So here is the bacteria, and we're only drawing the Lac Operon, a portion of its bacterial chromosome. So here we have looked at, we have three genes. The first gene is Lac C, which give rise to beta-galactosidase. Second one is Lac Y, which give rise to lactose permease, and the third one turned out to be Lac A, which give rise to galactoside transacetylase. And all of these is under the control of one promoter element, which is known as lactose promoter. Now, several base pair upstream to that, another sequence is present, which is known as Lac I. Now, Lac I gene product is basically a repressor. So, what happens is Lac I is always producing a repressor. So, Lac I is giving rise to Lac I mRNA. Then this Lac I mRNA is transcribed, Lac I mRNA is translated, and that is giving rise to a particular repressor. So this is Lac repressor. Now, Lac repressor can bind to the promoter, can go and bind to the promoter. Now, nobody is there to prevent this repressor to bind from the promoter. Now, in order for the transcription of these genes to start from the Lac promoter, one important thing, which is RNA polymerase, need to bind to these particular promoter region. But the RNA pol cannot bind because already the promoter is blocked by the lactose repressor. So RNA pol actually cannot bind. As a result, the genes which are in the lactose operon genes, they cannot be transcribed. So as a result, the state of the lactose operon is off. Now we would look at, so we have looked at a situation when glucose is high. Now we'll go down and look at a situation when glucose is low, but at the same time lactose is very high. So at that situation, what happens? We again draw the lactose operon. Now here is the structural genes. Here are the Lac Operon genes. Here is the Lac promoter. Here is the Lac I. Now, definitely Lac I gene is always expressed. So definitely it would give rise to its mRNA, and the repressor is produced in abundance. Now what happens? Whenever glucose level is low and the lactose level is high, what happens is some amount of lactose is converted to allolactose by the activity of the enzyme beta-galactosidase. Now the question is, if the Lac operon is off, where does this beta-galactosidase come from? So I have to tell you that even if the operon is off, majorly off, in any system, the transcription is pretty noisy and the gene expression is never off totally. So there is some amount of baseline transcription happening even in the presence of glucose, and the product of that baseline transcription is few beta-galactosidase and few amount of permease. And at the first place, these permease channels is helping the lactose which is present outside to come inside the E. coli, and that lactose is converted to allolactose using the enzyme beta-galactosidase, which is produced due to a baseline transcription of these operon. Now allolactose would be binding to this Lac repressor. Now allolactose is an inhibitor of this Lac repressor. Now when allolactose is bound, this Lac repressor cannot bind to the promoter region. As a result, what happens? Lac repressor cannot bind to the promoter, and as a result, our RNA polymerase can easily go and bind to the promoter, and it can give rise to transcription. And as a result, all the Lac Operon genes are transcribed and they form the necessary enzymes for lactose metabolism. So thereby we can understand that when lactose is high, the operon is not anymore suppressed. Now the operon and the suppression on the operon is removed. As a result, the operon is expressing. So we can, from these two discussion points, we can tell that the default state of this operon is off, because if you have good amount of glucose in the environment, why do you need even lactose? So the default state is off. Now what happens? The operon is only on when there is high lactose. Only lactose but no glucose. Right? As a result, the operon is turned on. So the default state is off. Now whenever on demand, whenever there is a metabolic demand of metabolizing lactose, the operon is switched on. So this kind of operon is an example of inducible operon. So Lac operon is an inducible operon because Lac operon by default it is off, but it is induced when lactose is present in the environment in order to metabolize the lactose. Now we look at a complicated situation. This complicated situation is now in the environment both glucose plus lactose is present. Now here you have both glucose and lactose. Now the question is, both the options for the bacteria is present. Now it have to choose between these two nutrients, and what the end result is, the bacteria would end up choosing glucose over lactose. They would prefer glucose than lactose because glucose is easy to metabolize than lactose. And so in order to do that, the bacteria need to devise a way to switch off the operon. Under the condition of both glucose and lactose, how the operon is switched off, that we are going to take a look at right now. So here we can understand that whenever both glucose and lactose is present, they would still prefer the glucose. So Lac operon is switched off or remained switched off in this situation. Now the question is how Lac operon is switched off. But this phenomena, when both the metabolite is present, glucose and lactose, that situation they are preferring glucose. This phenomena is particularly known as catabolic repression. So what is catabolic repression? So you can understand the catabolism of lactose into glucose is repressed because there is a lot of glucose in the environment, and that's why you don't need to break down lactose. So that's why the catabolism of lactose is repressed in presence of simple nutrients. This is known as catabolic repression. But we have to understand that how the Lac operon would be functioning in presence of both glucose and lactose. Now simply Lac operon functioning is not so simple, just like I taught you right now. So we go back to that concept and we would add one more player. So definitely when glucose level is high, so we first go to a situation when glucose level is high. So when glucose is high, always, since its transport is coupled with cyclic AMP generation sometimes, so cyclic AMP level is low. Now lactose, in order for this RNA polymerase to be optimally recruited into the promoter, it requires one important protein known as CAP, or catabolite activator protein. Now this catabolite activator protein binds to cyclic AMP. So it gets activated when it binds to cyclic AMP. Now in presence of glucose, the cyclic AMP level is low. So this is your situation 1. And since the catabolite activator protein activity is low, it cannot optimally recruit the RNA polymerase. Apart from that, we have also seen in absence of lactose, the Lac repressor is binding the promoter, occupying the promoter, and not letting the RNA polymerase to bind to that promoter. As a result, there are 2 situations which is preventing the RNA polymerase to be recruited onto the promoter and transcribe the downstream gene in presence of glucose. So catabolite activator protein is the second player. We look at a situation when glucose level is low but lactose level is high. So whenever glucose level is low, your cyclic AMP levels are high. Whenever the cyclic AMP level is high, your CAP proteins are highly active. Now whenever CAP is active, it gives rise to a positive impact and help the polymerase to be recruited onto the promoter. At the same time, we have seen that in presence of lactose but not glucose, lactose is converted to allolactose, and allolactose represses the repressor. As a result, repressor cannot occupy the promoter, and also CAP is helping to recruit the RNA polymerase in the promoter. As an end result, polymerase is recruited onto the promoter, and the Lac Operon genes could be transcribed, and the bacteria can produce the necessary enzymes for lactose metabolism. Now we go to the complicated situation when the lactose operon is still off, but in both glucose and lactose is present, and we try to look at why. Now in presence of both glucose and lactose, here we draw our Lac operon. We have our regular structural genes, which are Lac Z, Lac Y, Lac A, under the control of our Lac promoter. Here is our Lac inducer, which is forming the repressor molecule. Now it's forming the repressor. Now definitely we can understand when lactose is present, some amount of lactose is going to get converted into allolactose, right? Now that allolactose would definitely go and bind to this repressor, and as a result, this repressor cannot bind to the Lac promoter. So you might think that, okay, now the Lac operon is having a green signal, it is ready to go. But at the same time, you have to understand, when glucose is present, the cyclic AMP is low. As a result, the catabolite activator protein is not optimally activated. So catabolic activator protein is not optimally activated. As a result, the RNA polymerase that should be recruited into the promoter in order to start the transcription cannot get optimally recruited in when the catabolite activator protein is low. So here again there is 2 factors. One factor is allolactose is produced because lactose is present, that is repressing the repressor. Now repressing the repressor would free the promoter from the repression of the repressor, but the problem is the RNA pol cannot be optimally recruited because the catabolite activator protein is not active. Now as a result, the transcription from these genes would be prevented. Now no genes are transcribed, the enzymes are not there. So in this situation, as a nutrient choice, the bacteria would use the glucose rather than the lactose. So now the phenomena of catabolic repression is expressed in terms of that. So I hope this video was informative. If you like this video, give it a quick thumbs up and don't forget to like, share and subscribe our channel. Thank you.